Plants do not have lungs, yet they are breathing and producing energy. Guess how? By the process of respiration. Hello everyone. In this video, we are starting with respiration in plants. We have 10 subtopics starting with intro, then moving forward with glycolysis, fermentation, aerobic respiration, Krebs cycle, electron transport chain, oxidative phosphorylation, balance sheet, amphibolic pathway, and last is respiratory quotient. For an intro, we know that all plants are generally autotrophic as they prepare their own food by the process of photosynthesis. The breaking of carbon-carbon bond of a complex compound through the oxidation within the cell leading to release of energy, a minimum amount of energy is called the respiration. Also, ATPs, which means adenosine triphosphate, act as energy currency of the living cell. Here is the overall summary of the process. That overall reaction is we have glucose combined with oxygen in presence of enzymes which comes to release of carbon dioxide, water and 686 kilocalories of energy. Before going for the second topic, glycolysis, we have the important question that how do plants breathe? Plants, unlike animals, they do not have any specific organs for gaseous exchange is absent. So there are several reasons why. So number one, each part takes care of its own. Gaseous exchange. There is very little transport of gas from one part to the another part. Then second, plants do not present great demand for gaseous exchange, as well as root, stem and leaves respire at very far lower rate than we animals do. Yet for the photosynthesis, a large volume of gaseous exchange is required, which is provided by the stomata. Now let's move on to our first reaction or process, glycolysis. Glycolysis was discovered by three German scientists named as EMP, that is Emden, Meyerhoff and Paris. So these are scientist names. First, we are looking for theory part. So glycolysis has originated from Greek words glyca for sugar and lysis for splitting. Embedded Meyerhoff and Parnas developed the EMP pathway, a scheme for glycolysis. Glycolysis is a process in all living organisms involve partial oxidation of glucose in two molecules of pyruvic acid in a cell cytoplasm. So here we see, so glycolysis is a universal process. It happens in both of the organisms, aerobic and anaerobic organism, all of the living organisms, prokaryotes and eukaryotes. It is a partial oxidation of glucose and occurs on simple sugars only. That means glucose and fructose. We cannot proceed glycolysis process with sucrose or starch. Those are complex sugars. We need to proceed with simple sugars. This glycolysis process has two phases. First is energy consuming phase and second is energy forming or payoff phase. So first 1 to 3rd are energy consumed, where we give ATPs and rest of all we get ATPs as a byproduct. Also, the first, third and 10th step of the glycolysis is irreversible, which means they are irreversible, which means they are rate limiting reactions or rate limiting step of the reaction. So let's see the 10th step process of glycolysis. So first, starting with glucose, a simple sugar which is 6 carbon. So at first, glucose is converted into glucose 6-phosphate by addition of one inorganic phosphate from ATP, which is converted into ADP. Here hexokinase enzyme is used. Then we do isomerization of this glucose 6-phosphate into fructose 6-phosphate with the help of enzyme isomerase. Moving forward, we add another inorganic phosphate by breaking ATP into ADP and produced fructose 1,6-bisphosphate in the presence of phosphofructokinase. So this is first step and this is third step. Okay. Then from this fructose 1,6-bisphosphate, it breakdowns into triose phosphate but adds 3,3 carbon compound. First is glyceraldehyde 3-phosphate and second is dihydroxyacetone phosphate. These both are 3 carbon compounds which are isomers of each other and can be interconvertible. So first, and aldolase is an enzyme which breaks the fructose 1,6-phosphate into these two compounds. Then for further reaction, this dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. Then for sixth step, we convert triose phosphate by addition of two inorganic phosphates, but here we don't use ATPs. Instead of, we get inorganic phosphate from the compounds like H3PO4. Also, here 2 NADH are converted into 2 NADH plus H plus ion in the presence of PGL dehydrogenase. From the name of enzyme and conversion of NADH into NADH2, we can say that sixth step of glycolysis is dehydrogenation step, where glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate acid. This acid now moving further in 7th step, 2 ADPs are converted into 2 ATPs and we get 3-phosphoglyceric acid. In 8th step, moving with 2-phosphoglyceric acid or glycerate. Then in the 9th step, 2-phosphoglycerate is converted into 2. This 2 is number phosphophenol pyruvate, and at last 2 ADPs are converted into 2 ATPs with the help of pyruvate kinase. We get 2 molecules of pyruvic acid, which is 3 carbon compound. So for a net gain of ATP from one glucose molecule, we get direct ATPs 4 by the substrate level phosphorylation in ninth step, 9th to 10th, and in 6th step. Also, at the beginning, we lost 2 ATPs, or 2 ATPs are consumed. So, the net result of glycolysis is 2 ATPs, 2 NADH2 and 2 pyruvic acid. Also, for a note, phosphofructokinase is an enzyme which is called the pacemaker enzyme and step. Moving forward to fermentation. Fermentation by yeast completes glucose oxidation under anaerobic condition through reaction converting pyruvic acid to carbon dioxide and ethanol, catalyzed by enzyme pyruvic acid decarboxylase and alcohol decarboxylase. So note, less than 7% of the energy in glucose is released out. So glucose is converted into carbon dioxide and ethanol. If we see the process, glucose, glycerol dehydrate 3-phosphate, glycerol aldehyde 3-phosphate, getting NADH2, then converted into 3-phosphoglycerine acid, 3-phosphoglyceric acid, then phosphoenol pyruvic acid, which brings to pyruvic acid, then by pyruvic acid either converted into lactic acid or ethanol and carbon dioxide. If we see alcohol fermentation, the pyruvate or pyruvic acid, which is have formula CH3COCOOH, that is 3 carbon compound in 2 carbon, 2 pyruvic acid, in presence of Mg2 plus and pyruvate decarboxylase, we get 2 molecules of carbon dioxide and 2 molecules of 2 carbon compound CH3COH. On further reduction with alcohol dehydrogenase and by converting 2 NADH2 into 2 NAD plus, we get 2 molecules of ethanol, that is 2 carbon compound. In lactic acid fermentation, 2 pyruvic acid in presence of lactate dehydrogenase reduction process 2 NADH2 converted into 2 NAD plus, giving 2 molecules of lactic acid. Moving forward, for aerobic respiration. Aerobic respiration is the process in which there is a complete oxidation of organic substance in presence of oxygen and release of carbon dioxide, water and large amount of energy present in the substrate. This is theory part. So aerobic respiration starts with glycolysis, then middle is link reaction, then Krebs cycle and it is. So for now we are going to see the link reaction. So in link reaction, the pyruvate, that is 3-carbon compound, combines with coenzyme A in presence of pyruvate decarboxylate and pyruvate dehydrogenase by conversion of 2 NADH positive into 2 NADH2, we get acetyl coenzyme A and 2 molecules of carbon dioxide. This pyruvate decarboxylase is not an enzyme, it is a proper complex which contains TPP, that is thymine pyrophosphate Mg2 plus and lipoic acid. Moving forward to Krebs reaction or tricarboxylic acid cycle. The tricarboxylic acid cycle or Krebs cycle was proposed by Adolf Krebs in 1937. Krebs cycle operates in the mitochondrial matrix because it contains complex mixture of various respiratory enzymes. Starting with pyruvic acid or pyruvate, which is converted into acetyl coenzyme A in link reaction. This acetyl coenzyme A then enters in citric acid cycle, which is the another name of Krebs cycle. This acetyl coenzyme A enters in citric acid cycle and first enzyme citric synthesis, addition of H2 produced the citric acid, which is the first stable product of the reaction cycle and hence named as citric acid cycle. Moving further, this citric acid then converts to cis this citric acid then converts to cis anconidate, then isocitrate then oxalosuccinate, and comes to alpha ketoglucaric acid, which is 5 carbon compound. Then moving forward to formation of 1 GTB and NADH2. Here we also get 1 NADH2. Alpha ketoglutaric acid is converted into succinyl coenzyme A or succinic acid by the help of alpha ketoglutarate dehydrogenase complex. Then this succinic acid goes to form succinate and then fumarate, then malic acid, which is malate. While this we get in this step, we get one FADH2 molecule from malic acid or malate. In the presence of malate dehydrogenase, it is converted into oxaloacetic acid or oxaloacetate. While this we get one NADH2 molecule, and this cycle continues again for acetyl coenzyme A. So, if we write a balanced reaction of Krebs cycle, we get it as pyruvic acid plus 4 NAD plus FAD positive plus 2 H2 molecules and ADPs plus inorganic phosphate. In presence of mitochondrial matrix, we get 3 molecules of CO2, 4 NADH plus 4H plus FADH2 and ATP molecules. Krebs cycle provides the number of intermediates which are used in different anabolic pathways for important biomolecules like glutamate acid, aspartic acid, etc. Hence, this cycle is amphibolic in nature. Amphibolic pathway is the one which is used for both breakdown and build-up of reaction. All enzymes of TCA cycle located in mitochondrial matrix except succinate dehydrogenase that is located in inner mitochondrial membrane in eukaryotes only.